Hi, my name is Phil Smith, and I've been looking at the role of the evolution of sex with regards to aging and the resistance to aging. The evolution of sex is an old problem in biology. The main problem is why are there two sexes in most of the large organisms we see, vertebrates in particular, and why aren't they asexual? Asexuality makes more sense. You have twice as many offspring that can produce offspring. So you would expect that an asexual mutant would invade a sexual species and wipe it out. This doesn't seem to happen. The other thing you have to deal with is recombinational load. Just as recombination can bring unique gene combinations into being through recombination, it can also break up perfectly good functioning combinations. So as shown here, you have to show how you can pay for the cost of recombinational load. So that the benefits of recombination are not wiped out by the effects of recombination and the breakup of helpful combinations of genes. Neutral networks are networks of genomes that have all the same fitness values. So you only have two fitness types, viable and non-viable. So it's a binary fitness function. However, genomes do differ in their connectivity. Some genomes are in regions of genome space that have a lot of neighbours, and some are in sparse regions of genome space. And mutation can drive the population into better connected areas because those areas have a lower genetic load. You need some way of deciding which of, whether a genome is viable or non-viable, and some have used RNA folding or models of protein folding. And it's been shown that recombination is better at concentrating the population into the middle of the network. So the method I developed for generating my sets of viable and non-viable genomes was a modified version of the Wolfram elemental cellular automata. The reason I used this is they're well understood, and there's a range of rules that we could evaluate, and we could evaluate a range of different bit lengths. Here I show how we modified the Wolfram ECAs to accept or reject strings. Here we have rule 26, which gets its number from 2 to the 1, which is 2, plus 2 to the 3, which is 8, plus 2 to the 4, which is 16. It accepts 3 bit blocks that add up to 4, integer value 4, integer value 3, and integer value 1. You scan from left to right, it doesn't make any difference. Take the first 3 bits. These 3 bits are add up to 5, and that's not in our set, so we reject it. 2, that's not in our set either, so that's a 0 in the next line, or a white square. 5 again, it's not in our set, accept, so we reject it. 3, that's accepted, and 6, that's rejected. So then we scan the next line. That's 0, 0, 0 equals 0, reject. 0, 0, 1, it's a 1, so accept. And 0, 1, 0, that equals 2, so that's rejected. Then the last 3-bit line is 0, 1, 0, that's 2, so that's rejected. So now the whole string is rejected. So here's an example of 2 different bit strings. One's been accepted and the other one's been rejected, or both processed with rule 26. So for a given bit string length, we can apply this rule to every genome, and we can see whether that genome is accepted or rejected. So here we can show for a 5-bit string all the possible rules and all the possible combinations of genomes. Black shows that the string was accepted, and white shows that it was rejected. On the right hand side we have the same but with 9 bits, and a similar pattern but a bit more detail there, showing the accepted and rejected strings. Once we've got our set of genomes for our neutral network, we can, for small genome lengths where there's fewer possible genomes, we can hook all the genomes up to the nearest neighbours that are genomes but are only 1 Hamming distance away. And for the different rules we get different networks, and you can see some of them here. There's 256 rules, but a lot of the rules have left-right homologs and black-white homologs, so we don't need to look at all of the rules. There's more rules than there are here, but this is just a sample of some of them. So we can breed these populations of genomes that we generate using a given modified Wolfram rule. Here we can see the results of a breeding experiment that was 500 generations long. We started with a 51-bit genome. We randomly chose 5,000 genomes for the start population. Then, using asexual reproduction alone with a mutation rate so that each genome mutates every generation, you can see the reduction in load during this first phase of asexual reproduction. Then we switched at around about 150 generations to sexual reproduction. So there's recombination. We choose two genomes, recombine them, and choose the offspring, and then see if that survives. And we have a very high level of recombinational load. These are the black dots. So asexual is white open circles. Black dots are sexual reproduction. We see this drop in recombinational load that eventually plateaus out at equilibrium around about 350 generations. And then we switch off recombination, go back to asexual recombination. We see that the genetic load, or mutational load, is now lower than it was before we had sex. And so this low load is what's important, and you can see it gradually increases up back to the equilibrium level that it had before we had sexual reproduction. We can superimpose the Wolfram ECAs used to calculate the viability of every genome in a population, and by superimposing them on each other we can see the emergence of gliders at various stages of the evolutionary process. In this figure we see the start population, the population at equilibrium during asexual reproduction only, the population at the end of sexual reproduction, and the population that returns back to equilibrium under asexual reproduction. In this little video we're going to show the evolution of these states using a 51-bit genome using rule 124, and we're going to look at the changes in load under sexual and asexual reproduction, and the changes in the activity time portrait in those different regimes and how that changes over time, and also the frequency distribution of load. So that is how many of the genomes in the population can survive with have zero mutations that are lethal, how many have one mutation that are lethal. So we sample the genome at every bit and flip it and see if it's lethal, and count how many of those of the bits in the whole genome are lethal if the bit is flipped. We start off with asexual reproduction alone. The ATP pattern in the bottom right hand box is shaped by mutational load, as is the frequency distribution of the lethal mutations in the top right hand box. This is response only to mutation selection, so it's reduction in genetic load through formation of gliders. Next we introduce sexual reproduction. Initially the pattern in the ATP plot in the lower right hand becomes more diffuse, but as the reduction in recombinational load in the top left diagram continues, that pattern becomes clearer and clearer, and we see a shift to the left in the frequency distribution of load in the top right hand diagram. This continues until we reach equilibrium, which happens about now. Now we turn sexual recombination off, and you'll see that the load is lower than it was previously before we turn sex on, and is gradually increasing. The pattern in the ATP is becoming more and more diffuse as we go back to the mutation equilibrium level, and the frequency distribution of load is moving back to the right. Here we'll just repeat that experiment a few times, and you can see the movement of load to the left under recombinational load, and also a little bit under asexual mutation load, and the emergence of patterns in the ATP, which diffuse under mutation alone. You see it appearing again here. Should appear. You would get the mutational load pattern, and then recombination goes diffuse, and then it starts to get more and more intense as the population moves to the left in the frequency distribution diagram, and here it goes diffuse again under mutation alone. Rule 110 is an interesting rule in the Wolfram ECAs. It is Turing complete, and it forms a alphabet of gliders that can interact constructively and destructively to form new gliders and or new persistent structures. And if you look at its homologs 1, 2, 4, which is the left-right homolog of 110 and 137, they have very similar behaviors. Not all rules respond positively to recombination. Some rules have a negative response. The mutational load after recombination is actually higher than it was at mutation equilibrium alone. One of these rules is Rule 30, which is known to be a pseudo-random number generator, and if you compare it to using SHA-256, which is a random number generator to accept or reject genomes, it gives a very similar pattern, and that is that there is some decrease in load under mutation, but it is completely lost during recombination. The figure here shows on the top right hand side the negative response to recombination, and the bottom left is a positive response to recombination, and the top left is SHA-256's response to recombination. And you'll see the two, top left and top right, are quite similar in some ways. So it appears that mutation will move a population towards the center of a neutral network where there is less mutational load because the nodes are better connected. However, recombination seems to be more effective. It can squeeze it even into a more densely connected part of the node more effectively than mutation alone. This basically decreases the entropy of the genome, and therefore it can absorb more randomness before it fails. So this should have an effect on aging. So to model aging, we started with an organism with a fixed number of cells, and we just did a random walk for each cell from the population, either a sexual population or asexual population, and randomly walked if it had a lethal mutation and its cell died. If it was non-lethal, it just kept walking until all the cells were dead, and we basically just took the time for the organism to die as an indicator of resistance to age. Here you can see the difference in response to aging in the two different responses to recombination. The graph on the left shows Rule 129 at 21 bits, which is appears to be a random number generator, and you can see the sexual individuals in the dark bars. They died earlier than the asexual individuals, and on the right hand graph we have Rule 84, which responds positively to recombination, and the aging mortality is shifted to the right, so they are living longer than the asexual individuals under the same mutation accumulation rate. Note that aging here is linear, whereas in reality it's an exponential curve, so mutation rate is held constant. It may actually increase exponentially. So it appears that sex benefits longevity, and we can see some way in which we can pay the twofold cost of sex if we have an organism with late maturation. Late maturation means that the longer they live, the longer their reproductive potential. And you can see in the graph on the left we have the maturation age, which is the vertical orange line, we can slide that left or right and calculate the ratio of the two areas for the survival of the sexual species and the asexual reproductive method. And those two, that ratio of those two areas is the advantage of sex over asex. And you can see in the graph to the right, which shows the effect of moving that maturation age left and right, we get quite a significant boost in the reproductive advantage of being sexual, the reproductive potential of a sexual organism, because they live longer and are reproductive for longer. And we can get more, well over a twofold improvement at mutation times of around 30 to 50 mutation steps. It should be pointed out that this is a linear mutation, a linear aging model, so that graph may be distorted if we went to an exponential mutation accumulation rate. So, is there any data to back up this idea? Well, there's an old paper here by Burt, Austin, and Bell about the relationship between excess chiasma at meiosis and age to maturity. And you can see the graph above. There's a nice graph for mammals showing this relationship. There's some outliers, which is the vertical group of species, which I've highlighted there, which are dog, sheep, cat, goat, and there's down the bottom number 20, 28 and 27, I think, horse and bull. And you can see those groups are all the domesticated animals, and we think we know anyway that when an animal undergoes selection, the chiasma frequency increases. So this is probably what we're seeing here, and also there may also be inadvertent selection for longevity in domesticated animals due to the increased reproductive success of long-living stud animals. That's about wraps it up. So if you've got any comments or any feedback, put it in the comments below, and I'll put a link to the original paper below as well. And hope you enjoyed it, and good night.